In Chapter 2, we got a really nice introduction and basic foundation for chemistry. We're able now to move on to Chapter 4 that focuses on essentially organic chemistry, or chemistry involving those compounds that are carbon-based. So we know that living organisms consist mostly of carbon-based compounds, and there's a really good reason for this. Carbon, as you'll recall, has a valence of 4. It has a bonding capacity of 4, meaning it can form up to 4 covalent bonds with other molecular components or atoms, much like the model I'm holding here or the image that's shown in the bottom right-hand corner of this slide. Because carbon can form those 4 bonds, it's really useful for building very complex and varied molecules. These molecules include proteins, includes DNA, carbohydrates, and even lipids, which are all biologically relevant molecules. We'll spend some time discussing those in a later chapter. Now, again, by definition, organic chemistry is the study of compounds that contain carbon. Organic compounds can be really small. We're going to talk about some of the simplest organic molecules in the next few slides, but it can also include those molecules that are huge and carbon-based, like your proteins and your DNA. Now, most organic compounds, not only do they just contain carbon, but they also contain hydrogen atoms. And so it is a little bit of a discussion within the scientific community whether organic chemistry is limited just to those molecules that have carbons and hydrogens, or if we'll just include any carbon-based molecules, irregardless of whether they have hydrogens along with the carbons. Now, back in the day, back when we didn't have a lot of information about how life on our planet perhaps started, we definitely, as a scientific community, subscribed to this idea of vitalism, or a belief in a life force that's outside of any jurisdiction of physical and chemical laws on our planet. Pretty much what that means is we couldn't explain life. We didn't have an answer for why living things were alive, and so we just consider it to be kind of magical. And yes, while life is very magical, we have now shifted away from this idea of vitalism. We have some better ideas simply because of maybe even some accidental experiments that were performed, particularly by chemists. But before we move to that, right, when we did subscribe to this idea of vitalism, it was really thought that organic compounds, these carbon-based compounds that were found in living organisms, could only be produced by living organisms because that they contain whatever that magic that it was that allowed for those carbon-based compounds to be produced. And so the first sort of data to help us reject this idea of vitalism came from actually some chemists, particularly one chemist, a German chemist, who was trying to make an inorganic salt at the bench in his lab and ended up in an abiotic system producing urea, which is one of the components in urine that living organisms can produce. Another scientist that contributed his work to helping disprove the idea of vitalism was a man by the name of Stanley Miller. And his experiments are pretty famous. What he tried to do was replicate the environment on our planet before life existed to see if he could produce any organic based molecules without life, and just to get a better idea of how maybe life did start on our planet. So on the right-hand side, you see the contraption that he put together for his experiments to simulate the environment found on our planet before life existed. So down here, you've got the sea. So he's got a round bottom flask filled with water. He heats that water to allow some of that water to become water vapor, and he sends that water vapor up here to the quote-unquote atmosphere. Now, he probably didn't get the composition of the atmosphere quite right, but he did make a point with his experiment. So what he had put into the atmosphere were some methane, CH4, we have some ammonia, NH3, some hydrogen gas, obviously the water vapor in there as well, and then he had put some electrodes in there to introduce a spark. Now, those sparks likely came from maybe storms on the planet producing lightning. This could also be static electricity between some of the ash clouds that were the result of volcanic activity on the planet. Regardless, those sparks produced the necessary push to start chemical reactions in the atmosphere, allowing the atoms and the molecules up there to become rearranged to form new molecules, aka chemical reactions. And so Stanley Miller allowed his atmosphere to sort of exist for a little while, to go through those chemical reactions initiated by the sparks from the electrodes, and then he would allow the water to condense, and as it condensed, it would fall back through and be sent back to the ocean, but not before he would collect a sample for analysis to see what kinds of molecules were produced in the atmosphere after those chemical reactions, and then were dragged down by the water as it condensed. And what he found was that he could actually produce in an abiotic system in his atmosphere some amino acids that are important for living organisms on the planet. And so Stanley Miller and the German scientists, they were these pioneers of organic chemistry that helped to shift our view from vitalism to something called mechanism. Mechanism is the opposite, so it's the view that physical and chemical laws still govern all natural phenomena on our planet. So it's a little bit of history. Let's go ahead now, and before we build these carbon-based or organic molecules, let's review carbon for a minute. So if you'll recall from chapter 2, electron configuration is going to determine the kinds and the number of bonds an atom will form with other atoms. So if we, again, review carbon really quickly. Right, carbon is our 6th element, so that means that carbon has an atomic number of 6. It's got 6 protons, 6 electrons. So if we draw the atomic nucleus, we're going to start filling electron shells with those 6 electrons. We've got 2 electrons that go in the first shell. That one, remember, is full. So we need a second shell. We have 4 electrons remaining, 1, 2, 3, and 4. And there are your 6 electrons for carbon. Now, the valence shell for carbon here only has 4 electrons. It has a capacity of 8, so it would like to get 4 more. And so carbon is very happy to share 4 more electrons with other atoms to fill that valence shell. And so therefore it has a valence or a bonding capacity of 4, requiring those extra 4 electrons. And so this allows for really complex, very large molecules to be built using carbon. Now, as we start looking at carbon-based molecules, they have specific shapes. This is all a result of the way that those electron orbitals sort of interact with each other. But if we take a look at pretty simple organic molecules involving maybe just one carbon at a time, right? So we've got my model again. It's got the one black carbon in the middle, and 4 covalent bonds with other atoms or molecular components there. And you can see it has a very specific shape. It's a tetrahedron. And we can see this also in the first example on the next slide. So you've got your methane there. This is the simplest organic molecule. It's got 1 carbon in the middle, and then it's surrounded by 4 hydrogens to satisfy that need for those valence electrons for that center carbon. And again, you can see it's got that tetrahedral shape. Now, the tetrahedral shape is a theme that is continued when you start building even larger organic molecules. The second example here, ethane. Instead of having 1 carbon, it has 2 carbons that share a single covalent bond in between them. And you can see that there are 2 tetrahedrons, one on either side, one for each of the carbons, and they're flipped on their sides as those 2 carbons are bonded to each other. And that looks perhaps something like this. An interesting thing about when you have 2 carbons here in black that share that single covalent bond, there's a certain amount of spin to the molecule. So I can rotate one of the carbons and all the other things that are attached to that carbon around like that. Now we can get even more complicated. If we go back a slide, you don't necessarily have to have carbons sharing just a single covalent bond. Carbons can also share a double covalent bond. So if I take, for example, this molecule and allow these 2 carbons to share a double covalent bond, I'm going to have something that looks like this. So you've got your double covalent bond between those 2 carbons, and now you don't need as many atoms bonded to the carbons because the carbons satisfy their valence need with 2 electrons, with sharing 2 pairs of electrons, right, for a double bond. And so very similar to the model that I'm holding, we've got here ethene. So ethene is essentially the same thing as ethane, except instead of that single bond between the carbons, now you share that double bond. What's unique about the molecules that share that double bond between the carbons, the extra components, these 4 components that are attached to the carbons, they're all in the same plane. So you no longer have that tetrahedral shape when you have a double bond in the middle. Instead, it's more of a planar molecule in that particular spot where that double bond exists. The other thing to note is that there is no rotation. Having that double bond there prevents me from turning one of the carbons in respect to the other. So this means that it really matters what's on all 4 of these corners of the molecule because you can't rotate it. This will be something that'll come into play one more time when we start talking about isomers in later slides. So hold on to that thought. Okay. So we know that the electron configuration of carbon gives it covalent compatibility with many different elements, but carbon has its favorites. It really likes to form those covalent bonds with hydrogens, oxygens, and nitrogens. And this makes a lot of sense that these are going to be the most frequent binding partners for carbon based on their electron distribution, and therefore, it also makes sense that our bodies and organic, or let's say living organisms, are made primarily of those 4 elements, as we discussed earlier in chapter 2, right? Our bodies are mostly 96% made out of hydrogen, oxygen, nitrogen, and carbon, right? Simply because we're made out of carbon-based molecules, and nitrogen, oxygen, and hydrogen are the favorite binding partners for carbon. Okay, so carbon, right, is the basic atom that's used to build these biologically relevant molecules, and sometimes it bonds to hydrogen directly and sometimes it doesn't. On this slide, we're giving you 2 examples of molecules that are still highly relevant to living organisms, but that they don't necessarily have hydrogens directly attached to the carbons. So the top molecule is carbon dioxide, CO2. It doesn't have any carbons to begin with. It's just, it doesn't have any hydrogens to begin with. It does have a carbon in the middle, double bonded to each of 2 oxygen atoms. And this molecule, even though it doesn't have any hydrogens, it's still very relevant to living organisms. This is what we exhale as one of the byproducts of cellular respiration. And it's also, maybe even more importantly, the material that photosynthetic organisms use as their source of carbon to build organic molecules that we as consumers on the planet depend on for food. And then the bottom molecule is urea. Urea, again, is carbon-based, and it does have hydrogens. It's just that the hydrogens are not directly bound to the carbon itself, right? Instead, the hydrogens are bound to the nitrogen, but this is still an organic molecule, still a component of urine produced by some animals, and again, biologically relevant.